Good morning, everybody. Oh, let me put my microphone out. So I can. All right, let's try that again. Good morning, everybody. All looking forward to a good Wednesday. Me too. So just an FYI, I will not be here next Wednesday. I am traveling next Tuesday evening to the University of Montana. I'm giving a seminar next Wednesday, so that will occupy my full day. I will be in Missoula. Then I will be there much of the day on Thursday, then I will be back since we won't be officially meeting for lab on Thursday, so I won't be here for that either. And then I will be back Thursday night and back in here to see your bright, shiny faces on Friday morning. What that does mean, though, is I will probably produce a video lecture for you for Wednesday, so that we don't get behind. Okie dokie. Cool. I'm excited about giving. I'll get you a second, Jack. I'm excited about giving this seminar because it's the first time I'm actually going to get to tell an intellectual group, this will be mostly graduate students, about the complete picture of body heat management during flight in hummingbirds. It's been something I've been working on since 2010, and I've told little chunks of it, but this is the first time I get to tell the whole story, at least that much of it that we know right now, which is kind of fun. So I'm kind of looking forward to that. Yeah, what do you got?
So for a lab next Thursday, does that mean we just show up and do
No.
this? Or?
So after today, we will not formally meet for lab until the first Thursday in October. Okay. You guys are in project mode. Believe me, I get my pound of flesh out of you. So I'm totally not worried about the fact that you won't be in lab for 3 or 4 hours. That time is certainly available for anybody who needs to meet with me, except next Thursday. And some time is available for you to do experimentation if you want to do that. The lab is free for you to use for experimentation if you want to do that. But historically, in fact, there's never been an exception to this. When you guys design your experiments to do for the thyroid project, you will all do them outside of normal lab time. So your time commitment to this will be substantial, and I'm not going to make you come to lab just because we've got a scheduled lab time. So that's kind of the nature of this beast. Are there any other questions before we get started? You're all right on top of things, huh? Cool. Well then, why don't we talk about intros? So we got some fun stuff to talk about today. All righty. So when we last left our story, we were talking about optimal temperatures and optimal pHs, and how when an enzyme, because it's primarily protein, gets outside of its environmental comfort zone, we get conformational changes to that protein that alter the structure of the active site. And so it's going to have trouble binding to substrates, which means it's going to have trouble working. So enzymes are structured in a manner that allows them to function at least as close to optimally as possible within the particular environment they're designed to work. Okay, and so we use examples of digestive enzymes to illustrate that with regard to pH yesterday. That doesn't mean that environments always stay constant. Okay, you get variations up and down, but, you know, for the most part, within a very narrow range of conditions, or I should say, enzymes need to stay within a relatively narrow range of conditions in order to function well in a physiological system. There are a lot of other things that impact the functionality of an enzyme, and we're going to talk about a few of those today, assuming my little clicker works here. I am trying a new. No. Okay, maybe stick you up a little bit. Just do that. I'm trying a new connector today. My other one seemed to be having issues with HDMI. This one has no issues with HDMI, but hopefully it's not going to keep my clicker from working here. Okay, so I say in this bullet point some enzymes, but in all honesty, the vast majority of enzymes require some other atom or molecule in order to be functional. Okay, and we call these extra atoms or molecules cofactors. So these cofactors have to be bound in some way, shape, or form to the enzyme in order for the enzyme to work, in order for it to catalyze a reaction. Okay, that is not going to work. So we're going to fix that. Pull you out of there. Let's stick them on the other side and see if it'll work here. Stick it in this one. Let's see if that will. Isn't technology wonderful? Well, you know what they say about the best laid plans. Okay, so whenever we refer to the protein portion of an enzyme, we call that the apoenzyme. When we talk about an enzyme, staying close, this is like utterly ridiculous. When we talk about the enzyme plus its cofactor, we call it a holoenzyme. Okay? And so we look at this diagram right here, we see apoenzyme, protein portion, this cofactor. In this particular case, it's actually contributing to the active site before the substrate binds in there, bound to the protein portion of the enzyme, and then we call that a holoenzyme. Okay? And pretty much all enzymes, all the important enzymes that we talk about are going to involve cofactors. This is going to drive me like really crazy. Okay. You are all going to pardon me for a second because I'm going to try one more thing real quick. This will teach me to not try new technology out before I actually need it. Let's see if that works. Cofactors, they are divided up into two broad groups. Okay. The first group is what we call a coenzyme. So a coenzyme is a non-protein yet organic substance that is thermal stable, which means temperature changes don't bother it. And coenzymes themselves can be divided into two categories. If they are loosely attached, and an example of that would have been the hypothetical example I showed you in the last screen, where it's interfacing with the active site. Vitamins fall into this category. We call them a co-substrate. Okay. In fact, if you look at all these things, you've all heard the term micronutrient, right? The vast majority of micronutrients, if not all the micronutrients that you consume, as well as many of the supplements that you consume, are basically going to serve as cofactors to enzymes. That's why you consume them in these like ridiculously small amounts, because you don't need a lot. And that includes a lot of metal ions that are toxic in elevated concentrations. So anyway, co-substrates are loosely bound to enzymes, but there are some co-substrates, or some coenzymes, I should say, that are tightly bound to enzymes, and these are appropriately called prosthetic groups. They're firmly attached. An example of this would be biotin, which is a cofactor for pyruvate decarboxylates. If it's a prosthetic group, it's stuck on there and it's not coming off. Okay. So it's there for the long haul. A co-substrate could hop on, hop off. And often things like that are used to regulate enzyme activity. More on that in a bit. The second broad group of enzymes, or of cofactors, are what I refer to here as the metal ion activators, although there's some ions that you might not think of really as metals. And in this case, some are loosely bound and some are tightly bound for basically the same reason. Those that are loosely bound, and there are a bunch of them, okay, and here's a whole list of ions that serve as cofactors. Again, these things can be used to turn enzymes on or turn enzymes off, just by whether or not they're attached. So if you think of signaling systems, for example, you know, often an important second messenger in signaling systems is going to be something like calcium. Well, calcium's there to serve as a cofactor to an enzyme. Okay. Now, there are some that are tightly bound. And when they're tightly bound, I refer to these enzymes as metalloenzymes. And so hemoglobin, which does function as an enzyme, we'll talk more about that when we get to circulation later on, it's actually part of the molecular structure of hemoglobin. So it ain't coming off. Okay. Cytochromes are another example of that. Those key molecules that are part of the electron transport chain or photosynthesis. So there's a lot going on in terms of making enzymes active. Okay, they're interacting with a lot of other things, and as you might imagine by this point, this is a primary way in which your body physiologically regulates enzyme activity. You should start thinking about things like feedback right now. You can use cofactors as part of feedback reactions, because they can provide the limiting step. All right, questions? Here's just a table that lists out a whole bunch of ions and some various enzymes. You probably recognize a bunch of these. Just pick out a few. You know, zinc, you know, is necessary for alcohol dehydrogenase, taking hydrogens off of alcohols, or carbonic anhydrase, which is important for making bicarbonate. Okay. Protein kinase You ever learn about signaling systems? Well, you know all about protein kinases. All right, calcium, remember I just told you that. An important second messenger of many signaling systems. Okay, so what we're going to do now is we're going to list a lot of categories of enzymes. And these categories are based on the functionality of the enzyme, what they actually do. So the first one we've already talked about, the addition or removal of water. Hydrolases, okay? These include the esterases, carbohydrases, which we've talked about already, nucleases, deaminases, amidases, and proteases, which we've talked about. These enzymes add water to break a chemical bond. That's what they do. Okay, hydrases. Okay, fumarase, enolase, and so forth. Carbonated hydrase is an example of this. They will add water to a molecule without breaking a chemical bond. Okay, there are some enzymes that are responsible for transferring electrons. Maybe. There we go. Oxidases. You think of oxidation reduction reactions, okay? So that makes sense. Dehydrogenases. You think about the fact that electrons are often carried around as hydrogen ions. Think electron transport. Okay, a lot of enzymes will transfer a radical from one place to another. So transglycosidases, they move monosaccharides. Transphosphorylases or phosphomutases, they move phosphate groups. Transaminases move an amino group. Notice again how the names are very descriptive here. Transmethylases move a methyl group. And transacetylases move an acetyl group. Okay? Love it how these names make sense. Okay? There are enzymes that split carbon-carbon bonds. These are called desmolases. That perhaps is not quite so descriptive. You probably would guess that from the name. Changing geometry or structure of a molecule. What do you think that might be called? Huh? Yeah, isomerases. There you go. Cool. Okay. The joining of two molecules through hydrolysis of a phosphate bond in ATP or another triphosphate. Okay. Ligases, we all know about ligases. We talked about things like nucleic acid synthesis, DNA, RNA, they involve ligases. Okay. So those are just some examples of different categories of enzymes that are important physiologically. Okay, questions? I'm going to change topics. All righty. Let's talk a little bit about enzyme kinetics. So, Lenora Michaelis and Maud Menton. You know this happened a long time ago because her name is Maud. Okay? I don't think they name anybody Maud anymore. 1913, they came up with the first mathematical explanation or description of how enzymes work. You've probably talked about Michaelis-Menten equation, probably in Gen Bio, okay, as a descriptor of the relationship between the rate of an enzyme activity and substrate concentration. Well, they're the ones who did it. But the Michaelis-Menten equation is actually simplistic relative to the way a lot of enzymes work. So remember, we talked about the enzyme structure. We talked about how they all involve globular proteins or tertiary structure. But there are a whole bunch of enzymes out there that consist of subunits, multiple globular proteins, okay, quaternary structure. And these have a little different type of kinetics than simple enzymes that have just a single active site. And Briggs and Haldane, George Briggs and John Haldane, and we'll see John Haldane a couple times. He's a rather well-known old-time physiologist. We'll see him come up again when we get to the respiratory system. They re-derived Michaelis-Menten kinetics. Michaelis-Menten assumed this really rapid equilibrium between enzyme substrate and the enzyme substrate complex. It got there really fast. But that doesn't always happen. And so Briggs and Haldane reworked the kinetics so that that assumption was not necessary. Let's see what that looks like. So, hyperbolic kinetics, this one right here, this is standard Michaelis-Menten. You get a rapid rise to a Vmax when you've reached the maximum velocity of the reaction. This is typical of enzymes that only have a single binding site. So they have tertiary structure. One globular protein, one place to bind the substrate. Okay, that's what it will look like. However, enzymes that have multiple binding sites and typically exhibit quaternary structure, they exhibit what's called sigmoid kinetics. So this is Briggs-Haldane's version right here. It actually starts off very slowly in its rise to maximum velocity. And this is going to be doing, we'll talk about this more when we get to the end of this topic. This has to do with the fact that the binding sites on the different subunits of the enzyme, they interact with each other. We'll actually look at hemoglobin as an example of this when we get to the respiratory system. Okay. Any questions about this slide? Okay, let's walk through Michaelis-Menten kinetics. So for a lot of enzymes in a single binding site, this is the way it's going to look. And in fact, and I'll have a bullet point for this, but I'll say it now. When you look at, excuse me, at a lot of physiological reactions that involve some sort of a physiological substrate, you will find that they exhibit a very similar curve to this. And that happens because they're driven by enzymes. And so the rate at which these physiological processes work, and I'll give you an example, kind of a random example of this in a couple of slides. The rate at which these things work is governed by the rate at which the enzymes work. So they exhibit a similar curve. So characteristics. The maximal velocity, and they do reach maximal velocity, and the reason they do this is because there comes a point, even though this Michaelis-Menten curve here is a relationship between reaction rate, enzyme reaction rate, and substrate concentration, there comes a point when all the enzymes are working as hard as they can work. This is the point of saturation. All the enzymes are tied up with substrate. You're not going to increase the reaction by adding more substrate. At this point, you've reached Vmax. This is also good evidence for the existence of the enzyme substrate complex, because that is the limiting step. How fast can you make those? Well, once all the enzymes get saturated, you can't make them any faster. And so at that point, you've reached Vmax. The other key variable in Michaelis-Menten is this value of Km. Km is the substrate concentration at which you reach 1/2 Vmax. Now, something that you need to burn into your brains, because students get confused with this all the time. Vmax is a rate, but Km is a substrate concentration. A lot of times students will get confused and think Km is also a rate because it's tied to 1/2 Vmax, but it's the substrate concentration at which you achieve 1/2 Vmax. And when you do comparisons between enzymatic reaction, what do you think you look at more often, Vmax or Km? This is not an obvious answer. The answer to this is not obvious. I'm going to see if anybody comes up with it. Sure. When if I'm doing a study and I want to compare enzymatic reactions. So let's say I want to look at, enzymes that drive muscle activity, because I'm trying to look at running speed or something. And so I want to compare the activity of the enzymes. Am I more interested in looking at Vmax, or am I more interested in looking at Km? Which is the better variable for me to look at in doing that comparison? Let me set it up another way that may help you with this. Let's say I compared the Vmax between two enzymes that are related to muscle activity, and the Vmax is exactly the same. Yet, in one case, the muscles work slower. In another case, they work faster, but they have the same Vmax. What could cause that? Go ahead.
So you would want to look at Km because you would want to see how they react instead
Yes.
of the maximum number.
Yes, and I'll show you a slide of this in a minute. But there are a couple of advantages of looking at Km. First, Km is a single point on the curve. Is that true of Vmax? No, it's not. Once I hit Vmax, every substrate concentration beyond that is tied to Vmax. Yet I only have one substrate concentration that's tied to 1/2 Vmax. And so I can look at a single point. The other thing that Km tells you, and I absolutely will have a bullet point for this, but I'm going to tell you now because I'm excited about it. The value of Km can tell you something about the affinity of the enzyme for the substrate. If you have a high value of Km, is affinity high or low? Think about it. If I have to add a lot of substrate to a system to achieve 1/2 Vmax, what does that tell me about the affinity of that enzyme for the substrate? It's got a low affinity because I have to add more substrate in order to get to 1/2 Vmax. But if I have a lower Km, or if I have a lower substrate concentration to achieve Km, then the affinity is higher. And so if I'm looking at these two physiological systems, in our hypothetical example here, muscle function, maybe I achieve overall contractile force more quickly in one case because the enzyme in this case has a lower Km. And so it achieves Vmax more quickly than the other scenario. Does that make sense? So there's a lot you can learn by looking at Km. All right, here is the Michaelis-Menten equation right here. Yep, you do need to know it. You might even have to work it. But you want to know what? You are all such incredibly intelligent people. I will bet this simple algebra is not beyond your ability. Any questions about anything on this slide? Okay. So, just kind of review here, once all the enzyme molecules are saturated, you can't go any faster. Vmax is it. I already said this, high Km indicates an enzyme has a low affinity for a substrate. So there's my bullet point. Okay. So, big question is, are we still recording? Okay, touch screen to start. Well, that was good. I tell you, technology is treating us amazing today. You know, that never happened when I was drawing on a whiteboard. But of course, nobody could ever read anything that I drew on a whiteboard. Okay, there we go. So I need to, okay. Actually, it looks like I'm still recording, so I think we're good. Sure, everything is good here. Well, we're just going to trust that everything is good. Okay. In fact, I think I said everything on this slide. So we talked about the fact that Michaelis-Menten curves describe a lot of physiological reactions. Okay, so effect of substrate concentration. Okay. So we talked about how the Michaelis-Menten curve plots the enzymatic reaction rate as a function of substrate concentration. So that tells you that substrate concentration does impact the rate at which an enzymatic reaction goes. Up until you hit Vmax, then it doesn't anymore. But it does determine that you're going to get to Vmax. So substrate concentration does have an impact. But enzyme concentration has an impact on the rate at which a reaction is going to go as well. So for a given enzyme concentration, you have one value of Vmax. But one of the ways that you can make a reaction go faster is to increase the enzyme concentration. Because if you increase the enzyme concentration, then your Vmax value is going to be higher. That's illustrated in this slide right here. So right here, this orange line right here is a low enzyme concentration. So our enzymes get saturated at a lower level of substrate. And so Vmax is capped here about 2/3 up the y-axis. This blue line here represents a high enzyme scenario. So now you can process more substrate because you have more enzyme. And so your Vmax value is going to be higher. Does that make sense? So the way I want you to think about this is that from a physiological perspective, think about how a body can physiologically use things like cofactors to regulate the amount of enzyme that's active in the system depending on need. And think about how something like a feedback reaction might play a role in that. Does that make sense? So how enzymes work, it really fits well into the regulation of physiological function and the maintenance of homeostasis. So the rate of an enzyme reaction can be controlled either by substrate concentration or enzyme concentration. Here are just a couple of graphs which illustrate the impact of affinity. So on the left here, we have three curves that illustrate enzymes with variable levels of affinity. This low one here marked Z is a low affinity enzyme. The one marked X is a high affinity enzyme. And what you see here is that the slope of the rise to Vmax gets steeper as the affinity increases. If you look over here on the graph on your right, you have the low affinity enzyme and the high affinity enzyme, and you can see how the values for Km, that substrate concentration at which you achieve 1/2 Vmax, is substantially lower for the high affinity enzyme than it is for the low affinity enzyme. Okay, so this is a graphical illustration of what we've already talked about. Does this slide make sense to everybody? Okay. Did my PowerPoint drop out here? What's going on? I think it's because my health insurance was trying to call me, even though I told her I was teaching July. But that's okay. All right. I promised you a random example of a physiological process that exhibited a Michaelis-Menten curve. And so I just chose this one because everybody likes these funky organisms that are in deep sea hydrothermal vents, right? These are the giant tube worms here, and they function in these very deep sea environments where they have no light, because they have these symbiotic chemoautotrophic organisms that fix carbon for them down there, and basically replaces the process of photosynthesis. All right. And so when you look at a piece of data from this study, so we have the amount of carbon fixed by these symbiotic organisms, that are bacteria, and the CO2 concentration, the free CO2 concentration in the system, we see that it rises very much like a Michaelis-Menten curve and will eventually achieve a Vmax. And again, this is because it's rate limited by the amount of enzyme that's in the system. So this is just an example of how a physiological process exhibits Michaelis-Menten. Okay, now we have been looking at Michaelis-Menten curves and Briggs-Haldane curves, but a challenge of working with these curves is that they're curvilinear, which means you have to measure multiple points along that curve in order to plot the curve. You can plot Michaelis-Menten in a slightly different way. And you can actually do this with Briggs-Haldane as well, but it's most often done with Michaelis-Menten. And you can plot it as what we call a Lineweaver-Burke plot. And what the Lineweaver-Burke plot does is it linearizes the Michaelis-Menten curve. All right? So you can actually get the curve and calculate all of the Michaelis-Menten parameters with just 2 points. It's pretty cool. So the Lineweaver-Burke plot is a really valuable tool when you're trying to understand aspects of things like enzyme inhibition. We'll go through several scenarios of that in the next several slides. So This is a line we revert plot. Note it's a linear line. The y-axis is right here, it's plotted as to 1 over the velocity of the reaction. The x-axis is 1 over the substrate concentration. The slope is Km over V max. The y-intercept is 1 over V max. The x-intercept is 1 over Km. So it's the inverse of a lot of things. This here is the Langley-Revert equation. And if you look at it, you can see that it is basically an equation of the line. Y equals mx plus b. So m being the slope here, x being your independent variable, and b being your y-intercept. Okay. So one of the real values of the Langley-Revert plot is with just 2 points, I can tell you exactly what Vmax and Km are. Pretty useful, right? And there's lots of good ways where you can apply this sort of a relationship. So let's talk a little bit about enzyme inhibition. Enzyme inhibition is critical from a physiological perspective because physiologically, your own body uses enzyme inhibition to control reaction rates. So you make natural inhibitors of enzymes, or you have ways of inhibiting enzyme function. But we also use it pharmacologically. We make drugs that are specifically designed to inhibit enzymes to prevent what it is the enzymes are trying to make happen. And there are tons of examples of that. Okay. Studies that use inhibitors have told us a ton about the nature of enzyme active sites. A lot of what we know about active sites and how specific they are comes from designing things to fit into active sites and seeing how close they have to get before it will drive an enzymatic reaction. So in essence, you're building inhibitors. As I mentioned, many drugs use enzyme inhibition as their mode of action. And so we can prevent some disease states simply by inhibiting enzymes. So a protease inhibitor, ritonavir, which is a primary drug that has been used to treat HIV. It does this. Okay. It inhibits enzymes that break down proteins. Poisons will inhibit enzymes. So we're going to talk about different categories of inhibitors. Poisons can be big time inhibitors of enzymes. And I'll give you some examples of that. Enzyme inhibition, it can be reversible, which is the good kind of inhibition, or it can be irreversible, which is the bad kind of inhibition. I'll bet you can guess what kind poisons are. When we talk about reversible inhibition falls into 3 categories. It can be competitive, non-competitive, or a relatively new category called uncompetitive, which makes it sound neat. And each of these have distinctive characters that we can determine by looking at Langley-Revert plots. So, how do metabolic inhibitors work? Through enzyme inhibition. So here we have a hypothetical biochemical pathway. We start off with a substrate. We get an enzyme that converts it to a product. We get a second enzyme that converts that product to a second product. And a third enzyme that creates our final product out here. There are a couple of ways that you can use enzyme inhibition to control this reaction. First, you can just produce an inhibitor that will inhibit one of the enzymes in the pathway. In this case, they're illustrating an inhibition of E2, which will block this pathway, which means the process stops at the production of the first product. Alternatively, you can use feedback. So we've talked about feedback a couple of times. You can use this end product, P3, to feed back and inhibit the enzyme that drives the production of the first product. That will also stop the reaction, and it will stop it in a way that's dependent on the amount of P3 that's available. Have we looked at an example of this, a real life example of this yet? I wouldn't ask you if we had, right? What's an example of something like this? We've talked about. First off, is this positive or negative feedback? It's negative feedback, yes. Even though that was a very low volume model. So here's some notes starting to flip. An example of negative feedback that could fit this type of pattern.
I know, or if I remember correctly, it was related to thyroid function.
Actually, that is, yeah. Actually, thyroid hormone was an example. It's not actually what I was thinking about, but that does work. Yeah. What was the other one? Even before we talked about thyroid, we talked about thyroid when we were talking about multiple feedback mechanisms working together. It begins with a P. Yeah.
Is it the PRL,
Yeah, prolactin.
prolactin, and then it releases dopamine to?
Yeah. Yeah. Okay. All right. First type of inhibition, and we'll probably stop after this. Competitive inhibitors. Competitive inhibitors are inhibitors that have close structural similarity to substrates, but are different enough from the substrate that while they can bind to the active site, they cannot be converted to product. So basically, they clog up the active site of the enzyme. This type of inhibition is reversible. An inhibitor, an enzyme inhibitor complex is formed. And so what it does is it ties up that enzyme and reduces the reaction rate. Because that enzyme can no longer bind to a substrate because the inhibitor is in the way. Okay. You can overcome this type of inhibition by simply upping the substrate concentration. Because what you have is the inhibitor and the substrate competing, that's why they call it competitive inhibition, for the active site. So if you create a scenario where the substrate way outnumbers the inhibitor, then the effect of that inhibitor will be reduced. Because it's a pure probability thing. Does that make sense? All right. Let's see how Langley-Revert plays into it. Here is a Langley-Revert plot for competitive inhibition. We start off with no inhibitor on this lower curve here, high inhibitor on this upper curve on the right-hand side of the curve here. So inhibitors going up as you move up on the y-axis. Tell me what you see. What do you learn from this? Think in terms of Km and Vmax, the two primary variables associated with Michaelis Menten. Well, what's changing and what's not changing? Yes.
Does the Km value change, like the concentration changes so that it can outcompete like the inhibitor?
Yes. So in this graph, and since we're out of time, we'll speed through this. Vmax doesn't change, but Km changes. Km increases. And looking at this graph, remember that the axes are inverse values. Km increases as inhibitor concentration increases, but Vmax doesn't change. So what is competitive and competitive inhibitor mimic in terms of substrate affinity to an enzyme? When you get a high concentration of inhibitor, what does that mimic with regard to enzyme affinity to substrate? Well, what happens to Km when affinity goes down? Yeah. Yeah, it would mimic a lower affinity rate because you still get to Vmax. You still get there. But it takes more substrate to do it. So that is the same scenario you get when you get a lower affinity. So in essence, it's mimicking a lower enzyme affinity for a substrate. And so if you see this scenario, when you see this scenario, this tells you that it's competitive inhibition. Okay. On Wednesday, we'll talk about non-competitive inhibition and uncompetitive inhibition. Questions? If not, enjoy the Sunday. Long lasts. We've got no more power moves.